So I'll do my best to follow the quadfecta. So I'll talk about, and we're not going to talk too much about soybean actually in my talk. We're going to focus on a pathogen of soybean called Phakopsora pachyrhizi, which causes a disease called soybean rust. And over the last several years, my lab's been interested in understanding the effector molecules that are produced by soybean rust that could enter into soybean plants and cause changes to the plant immune system. So my talk today will consist of three main parts. I'll give a brief introduction to soybean Phakopsora pachyrhizi interactions. We'll look briefly about some bioinformatics that we did to sequence the hostorial transcriptome to look for secreted proteins, many of which we believe are potentially effectors encoded by the pathogen. And then I'll talk about screens we've been doing to discover effector-like functions amongst the secreted proteins that are expressed in Hostoria. All right, so just a brief introduction then to the pathosystem. So we have a field of soybean, and then under conditions that are conducive for the disease, you can have a very severe epidemics of soybean rust that can essentially defoliate the crop. And so we've been interested in understanding over the years, understanding the responses of soybean to soybean rust. And I just summarize some of the responses here, right? See, these are the main responses. Maybe a trifecta of responses. I don't know. So we have an immune response, which is conditioned by the RPP1 resistance gene. So isolates that are recognized by the RPP1 resistance gene cause this phenotype, which is basically you can't tell that anything's happened to the plants. If you have RPP genes 1b or 2 through 6, and if that isolate's recognized by one of these R genes, you get a red-brown type of lesion. I don't know. Yeah. You get a red-brown type of lesion, and inside this lesion you'll find that there's very little fungal growth and virtually no sporulation of the fungus. And over here we have tan lesions over here. So in the tan lesions, which is characteristic of a susceptible cultivar, you would find that there's a lot of fungal growth and a lot of sporulation, right? So in my lab, we've been interested in understanding what's the molecular basis for this recognition, and how is the pathogen doing things like suppressing host immunity to cause disease. And so one of the things that we've been interested in the last few years, as I said, is what's the suite or repertoire of effectors that are introduced from the fungus into soybean plants. And so this would be a cartoon of the fungus, which would be within about 48 hours after a spore lands on the surface of a leaf. All right, so spore has landed, it's germinated, it's penetrated this epidermal cell, and it started to form intracellular hyphae, and ultimately it forms this hostorium here. And then within what we know from other fungi, and this should be true for soybean rust as well, is that these fungi express proteins either in hyphae or in their hostoria that are secreted out of the fungus and taken up into the plant cell, or they may have activities actually outside of the plant cell as well. But these would be the effector molecules that we're interested in, or the effector proteins, okay? So, and in particular, we've been interested in the effector proteins that are produced by these hostoria, right? So hostoria have been shown to be an important point of communication between a plant cell and a fungus. And it's been shown that hostoria can secrete proteins which are then taken up into plant cells. And so some of the functions of these effectors that are taken up into plant cells are to actually suppress the plant immune system and promote the growth, which promotes the growth of the fungus. So in soybean rust, we're interested in identifying proteins with these activities. So as a starting point for getting into studying effectors in the soybean rust, you can isolate hostoria from infected tissues. So this shows a micrograph of Haustoria that had been isolated from heavily infected soybean plants at about 14 days after inoculation with Phakopsora pachyrhizi, right? So you see these haustorial bodies here, and then this kind of neck structure. So that seeing this neck on this body is kind of a giveaway that these are Haustoria. And so, so we isolated the Haustoria, and then we did a 454 sequencing and assembled this haustorial transcriptome, right? Well, did I say we extracted RNA, we made cDNA, and then we did the sequencing. Just to make that clear, we're sequencing the transcriptome here. Okay. There is still no genome sequence for this fungus. It's very complicated. All right. So we got the haustorial transcriptome, and after going through bioinformatics filters and things like that, we ended up with about 4,483 unique contigs. And then we ran these through additional bioinformatic filters, like we're interested in identifying proteins that have a signal peptide, which would indicate that they should be secreted out of the fungus, right? And we ended up with 189 contigs. And then we did some other analyses which would eliminate proteins that would be predicted to be retained within the fungal cell. And so we ended up with about 150 predicted secreted proteins that are expressed in Haustoria. And this is just this is a size distribution of these 150 or so proteins. And so we considered proteins that were greater than 50 amino acids up to whatever the biggest was, which was around 800 amino acids. And I've indicated in blue. These are the proteins that we targeted for cloning then, and to study their functions in more detail. So all the proteins from ranging in size from about 89 or 90 amino acids up to our largest proteins were cloned from the fungus, and then we plugged them into well, it's called a gateway cloning system. I don't want to get too deep into this, but anyway. What we did was we took those, there's like 82, we ended up cloning about 82 candidate effector proteins. What we did was we cloned the open reading frame minus the signal peptide that was predicted. All right, and this was cloned into what's called a gateway entry vector. And once we have a protein in a gateway entry vector, we can move them easily into any so-called gateway destination vector, okay? And so what we've done with our 82 candidate effector proteins, we call them PPECs, Phakopsora pachyrhizi effector candidates. We've put them into a bacterial expression vector, which I'll show you a little more in detail in a little bit, a yeast expression vector, a plant expression vector, which allows us to fuse a GFP, a green fluorescent protein tag. And then we've also put them into a potato virus X expression vector as well. All right, and then once we have them cloned in these vectors, then we can do various different kinds of assays and express them in different host or plants or in soybean or whatever. All right, so gateway system is very nice for rapidly moving things into various expression systems. And one expression system I wanted to focus on in the next few slides, but I wanted to give a little introduction to it, is we're taking advantage of a bacterial type 3 secretion system. All right, and the reason for this is that rusts are impossible to work with, right? So we can't genetically manipulate a rust. We can't make transgenic rust or anything like that. So what we need is we need to find other systems, heterologous systems, where we can express proteins and study their functions, because we can't do it with the rust itself. And so we turn to bacterial delivery systems. So we use Pseudomonas strains like Pseudomonas syringae, pathovar tomato, or Pseudomonas fluorescens. And these have what's called a type 3 secretion system. And so what we do is we fuse the type 3 secretion signal of the bacteria to our effector candidate. And then this allows the bacteria then to secrete the effector candidate out of the bacterium and into the plant cell, and we can study what happens when we introduce this effector candidate into a plant cell through this delivery system. All right, and so so we screened all 82 candidate effectors in this particular assay, where we simply expressed the effector candidates in Pseudomonas syringae pathovar tomato, and we looked to see if any of the 82 PPECs would suppress the hypersensitive response that's caused by Pseudomonas syringae in tobacco, soybean, or Nicotiana benthamiana. And so after screening the 82, we found one, which we called PPEC23. And you can see that here's the hypersensitive response that's induced by this bacterium in tobacco and in soybean and in Nicotiana benthamiana. But if you express PPEC23, you see that the hypersensitive response no longer occurs, right? So this effector candidate suppresses the HR in tobacco, in soybean, and in Nicotiana benthamiana. So this was our first clue that soybean rust is producing effectors that can suppress some aspect of the plant immunity. This one, PPC23, can suppress the hypersensitive response. Okay. We've done other types of different suppression screens. This one, we're looking for what's called a PAMP-triggered immunity suppression, or a basal defense response. And this is a little more complicated assay. But what we do is we infiltrate a leaf with this ethan strain of Pseudomonas fluorescens, which is a non-pathogen. But ethan induces something called a basal defense response called PAMP-triggered immunity, or PTI. All right? And this PTI basically prevents Pseudomonas syringae pathovar DC3000 from inducing an HR. So what we do is we infiltrate a leaf with both this ethan and this PST DC3000, and then we look at this overlapping region. And if there's no effector present, then you get no HR, right? Because PTI is active, and it's preventing PST DC3000 from doing its thing here. And here's showing you the real outcome. So you have HR over here with the PST DC3000. Here's ethan, and then in the overlapping region no HR. So what we're looking for, if PTI is suppressed, what we're looking for is that hypersensitive response will form in this overlapping region. And so I just have a few examples of kind of weak suppressors of PTI and strong suppressors of, kind of medium and then strong suppressors of PTI, right? So in total, we found 17 PPECs that will allow PST DC3000 to induce some HR to some extent in this overlapping area, right? So now we've expanded our list of effector candidates from soybean rust. So we have one that suppresses HR, and now we have 17 that suppress PAMP-triggered immunity in some form. And there are this is a different kind of assay that we did to also look at the suppression of this PAMP-triggered immunity. And this is we're looking at the deposition of a polymer called callose, okay? And I'm not showing you all the data, but just showing some examples here. So in this assay, we are working with a mutant of Pseudomonas syringae DC3000 called delta CEL, this is a bacteria again that's lacking some of its effectors. And so if you try to inject, in this case we're using Arabidopsis, so if you inject Arabidopsis with this delta CEL, you get very strong deposition of callose. And that's in contrast to PST DC3000, which is the parent of this delta CEL mutant. You get very little callose deposition. So we introduced the 82 PPECs into this delta CEL mutant. And what we were looking for was any PPEC that would reduce the amount of callose deposition caused by delta CEL. And so this just shows an example of 3 different effector candidates that reduce the callose deposition. So effector candidate 3, 16, and 49. Again, these are kind of representing different classes of reduction of callose deposition. So we have kind of weak suppressors of callose deposition and moderate, and then strong suppressors of callose deposition. And in total, we found 15 PPECs can suppress callose deposition to one extent or another. All right. So basically, 15 of the 17 PPCs, which I showed you on the previous slide, also suppress callose deposition. Okay, so that's just a brief look at some of the assays that we did to find these effector candidates. We've also used a yeast cell death quadfecta. We're working on the quadfecta of assays here. We have suppression of bax-induced cell death in yeast. So bax is a protein that, this is actually a mouse protein that suppresses cell death in mice, but it's also been shown to suppress cell death in plants and in fungi. And so we thought it could be interesting to screen our PPECs, and we can do this very rapidly in yeast, to see if any of them suppress bax-induced cell death in yeast. And so here's a positive control. This is a very strong suppressor of bax-induced cell death in yeast. And you can see the bacteria grow quite well, even at very low dilutions. And we have 16 of our PPCs also very strongly suppress bax-induced cell death, and some are moderate suppressors and some are weak suppressors. And then a few of them don't suppress bax-induced cell death at all. So we think that possibly, our assays that we've done so far in plants, we've uncovered some of the cell death or defense suppression functions, but the yeast kind of hints that there may be other PPCs that we should be looking at for suppression of immune or immune-like responses in plants. Here's a list of the size distribution of the yeast cell death suppressors. And one thing we saw is that there's a big bias in the ability to suppress cell death towards the smaller PPCs, and many of them have unknown functions when you look at their gene sequences. So I think there's a lot of interesting things to explore with these smaller PPCs that we have no idea, based on homology to other things, what they do. And then another thing that we've been interested in is looking to see if any PPECs will induce defense responses, so especially in other plant species. So the idea behind this is that there's really not a lot of good candidates for resistance genes yet in soybean to soybean rust, right? And so what we decided to do was to look into other plant species to see if we could maybe find a hint that there may be resistance genes that could be useful in other plants. And so we've screened tomato. So we've used agrobacterium to transiently express the PPECs in tomato. And we found one, which we call PPEC6, that induces a hypersensitive response in tomato leaves. But I think more interesting is some work we did with a Korean group that screened 2 different pepper cultivars with all 82 of the PPECs, and they used a potato virus X expression system. But they found that several of the PPECs can induce HR in these pepper cultivars. So this shows an example of a positive control for hypersensitive response in pepper. And then here's our negative GFP control. And then looking at a couple of the PPECs that induce HR. These 2 induce HR in both pepper cultivars. Okay. But in pepper, we found that there appears to be some cultivar-dependent responses to the PPECs, because we have 12 that uniquely induced HR in ECW30R, and 3 that uniquely induced HR in CM334. But I think if I was looking for a pepper resistance gene to transfer to soybean, I would look maybe at these, look at these, what's recognizing these 2 PPCs here. All right, and then just the last thing that we've been able to do, because we're not only interested in the basic functions and whether they suppress immunity or not, but we are also interested in where the PPCs might be functioning if they are actually entering into the cell. And so I mentioned that we used a GFP fusion construct, and we can express these GFP fusions in plants. And then we can look to see, using confocal microscopy, we can look to see where in the cell are these GFP fusions. And so we find 5 basic different, a quintessecta of patterns. So we have a distribution which is in the both nucleus and cytoplasm. We also have a nucleus and cytoplasm, but instead of having a nice even distribution in the cytoplasm, this one is an example where we have these so-called cytoplasmic bodies. I don't know what that means. We're just using the term bodies. Then we have PPCs that are cytoplasmic only, so they're not actually in the nucleus, but they're restricted to the cytoplasm. And then we have PPEC15, which I think is unique amongst our 82 in that it's only present in the nucleus. And we have PPEC10, which is it's in the nucleus, but it's if you look over here, you can see it very nicely. It's very specifically localized to the nucleolus, right? So it may have some function in the nucleolus. So we have some interesting distributions of these proteins which are secreted by a fungus, taken up by a plant cell, and then probably relocalized within the plant cell to wherever their destination is, where they'll carry out their functions. And then my last slide here, for the data, this just summarizes the main properties of the various PPECs that we found that had at least a PTI suppression function. So we're not going to go through all this. All right. So in summary, we've used a lot of screens involving especially heterologous systems like bacteria and viruses and agrobacterium to identify PPC proteins that affect plant immunity. Right. So we found one that suppresses the hypersensitive response or effector-triggered immunity. We found 17 that suppress basal defense responses. We found another 17, some overlap with this list, that activate immunity in pepper and 1 in tomato. And we found 5 different profiles for localization within the planta. But we know that we're not uncovering all the potential functions of these, we're not uncovering all of even the immune suppressors, because we think we have a bias towards effectors that target conserved functions, because a lot of our assays are not done in soybean. They're done in tobacco or Nicotiana benthamiana or Arabidopsis. So we think these 17 are interesting. They may be targeting conserved functions, but it might be interesting to follow up the others to see if we can identify what's special about or what's specific about the interaction between Phakopsora pachyrhizi and soybean. So I'll finish with just some of the future work that we're working on. Many of the effector candidates have no predicted functions, and so one of the entrances into studying these more is to identify some host proteins that they interact with. So we're following up some of this. We're focused on one effector candidate called PPC15. That's the nuclear localized one I showed you. We're working on determining the cleavage site. It's actually annotated as a protease, and we've been able to show that it is indeed a protease. And we're trying to determine what its cleavage site is, and then incorporate this into a resistance gene system that involves a protein called PBS1. So hopefully we can make some progress on this and potentially develop a new or a novel resistance specificity to soybean rust and soybean. We're also getting into effector-effector interactions. So we've thought a lot about individual effectors inside of plants, but in actuality, when a fungus infects a plant, it's introducing hundreds of effectors, right? So we're trying to think of ways where we can study what's happening when there are multiple effectors in a cell at one time. So we're starting to focus some effort on this. And then we're also doing some work to expand the Phakopsora pachyrhizi effector repertoire. So we know that there has to be more than about 150 effectors. For other fungi, for other rust pathogens, people predict up to 400 to 800 candidate effectors. So we think we've vastly underestimated it with our first transcriptome sequencing. So we're working on expanding this list. So with that, I'll end and thank people who are involved in this work. I should mention Ming-Xing Qi. Most of the work that I presented is Ming-Xing Qi's work. And thank the funding agencies here. All right.
Any questions?
Michelle, she's behind you.
I knew you did it on purpose. Yeah. So you targeted all the effectors that were over 90 amino acids,
Yeah.
but there's lots of things that say a lot of effectors are very small proteins.
Yeah.
Given your success now with all these different systems for characterizing them, are you going to go back and look at some of those smaller ones?
So the question is, we focused on larger effectors. Are we going to go back and look at the smaller ones? Maybe. So we're doing some work right now with more transcriptome sequences. And so we want to be more confident about making sure that small things are full length. So once we do that, I think we'll be more confident in going after some of the small stuff. Yeah. So, but definitely. Yeah.
So when you say the BBEC23 on Organization 2, did you also go ahead and say it on the rest of the Allelic series for the RPP candidates?
Actually, there's no point in doing that because we cloned PPC23 out of several different isolates of Phakopsora pachyrhizi, some of them which induce HR, some of them which are recognized by RPP genes, and there's no genetic diversity, so there's no correlation with amino acid changes that would cause something to be recognized by, say, RPP2 or 3 or 4. So we won't do it for that reason. Yeah. Yep.
Have you ever tried other PTI inducers apart from what is it?
Have we looked at other PTI inducers? So we tried to do some work with like flag, but the assay didn't work very well in our hands. So we're trying to I have some ideas about different types of PTI assays to do. So we're going to revisit that. Yeah.
They're using like HARC-C instead of just
Well, there's like dozens of assays you can do. So you you have to do things that are. So we're trying to work with 82 effector candidates at one time, right? So we got to do things that have a reasonable throughput and that kind of thing.
one more question.
Yeah.
So are you doing like transgenic Arabidopsis with these effectors?
So PPC23, we've made transgenic Arabidopsis and transgenic Benthamiana. And we see the same things in transgenic plants as we do in, like if we do a type 3 secretion injection or something, we get the same phenotypes. So yeah, we're quite confident things are happening inside of the plant cell.